Nggak ada suaranya, Non. No sound.
Can you hear the voice, the music?
No, no, no.
No? Oh, okay.
No music, yeah. Too light.
Sorry, Prof.
Kecil sekali.
Kecil.
Has yielded into a new approach. Because of that, USAID initiated One Health Workforce Fund, areas. Those are multi-sectoral engagement, education and training, and institutional strengthening. Since 2014, OHW project has been implemented in 2 continents in Africa, known as One Health Center in Eastern Africa, as OHW.
Nggak ada suaranya, beresek ya.
Indonesia plays an important role in global health. Indonesia One Health University Network, or known as IndoHU, is the frontline and pioneer in aggregating human capacity One Health workforce. Established in 2012, IndoHU focuses on fostering One Health collaboration with Asia, professions effort to combat emerging health problems in animals. Those are education and training.
Tidak ada suaranya ya.
In 2018, IndoHU formed OHCC Chiagrama University. And in 2019, IndoHU formed OHCC Chendrawansi.
Over the past several decades, many previously unknown human infectious diseases have emerged from animal reservoirs, including agents such as SARS coronavirus, human immunodeficiency virus, HIV, avian influenza, MERS coronavirus, Ebola virus, and others. The increasing interconnectivity between humans, animals, and the environment has yielded into a new approach. Because of that, USAID initiated One Health Workforce Project, or known as OHW, which has three key areas. Those are multi-sectoral engagement, education and training, and institutional strengthening. Since 2014, OHW project has been implemented in 2 continents in Africa, known as One Health Central and Eastern Africa as OHCEA, and in Asia, known as Southeast One Health University Network, or COHUN. The collaboration has spread across 162 faculties and 84 universities in 12 countries. As one of the biggest archipelago countries with vast diversity, Indonesia plays an important role in global health. Indonesia One Health University Network, or known as IndoHUN, is the frontline and pioneer in advocating human capacity One Health workforce. Established in 2012, IndoHUN focuses on fostering One Health collaboration in Indonesia that encourages multi-professions effort to combat emerging health problems in human, animal, and the environment. Stated in National Long-Term Development Plan, RPJP, our vision aligns perfectly with Indonesia's big vision, which is community empowerment through education and training. IndoHUN has 3 main focuses, and those are education and training, multi-sectoral engagement, and institutional strengthening. IndoHUN established a collaborative center at the university level called One Health Collaborating Center, or known as OHCC. OHCC was formed from 2015 to 2019 and located throughout strategic areas in Indonesia. In 2015, IndoHUN formed the first OHCC at Gadjah Mada University. In 2016, IndoHUN formed OHCC Udayana University. In 2017, IndoHUN formed OHCC Airlangga University. In 2018, IndoHUN formed OHCC Syakwala University. And in 2019, IndoHUN formed OHCC Cendrawasih University. From 2015 to 2019, a total of 52 educational programs, 27 community outreach programs, and 17 research studies have been conducted by OHCC. Until now, 3,622 people have participated in OHCC activities, and 24,197 people have benefited from the existence of OHCC. OHCC has also conducted various programs and collaborated with various institutions such as University of Montpellier, University of Tokyo, Hokkaido University, UNSW Australia, Nivea, Nikkei Diagnostic Center, OIE, CIRAD, Gryphon Scientific, and Veterinary Domain Public-Private Partnership. What we have done is not finished yet. There are still many tasks which need to be done to improve national and global health. We understand that it is a complex challenge and impossible to be solved alone. Through coordination and strengthening collaboration across sectors, disciplines, and countries, we can tackle global health problems and challenges together. Less me, more we. IndoHUN social media channels: Instagram @indohun.id, Twitter @indohun, Facebook, LinkedIn, YouTube, Indonesia One Health University Network. Website indohun.org. Over
disease called COVID-19 that is caused by severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2. As of 14th June 2020, over 7 million cases of COVID-19 have been reported from all over the world, causing more than 400,000 deaths. COVID-19 was first identified as an outbreak of respiratory disease cases in Wuhan City, Hubei Province, China. In the following months, it spread rapidly to other countries with cases doubling within a short amount of time. SARS-CoV-2 is a single-strand positive RNA virus. It belongs to the family Coronaviridae, as the name, with the characteristic crown-like spikes on their surfaces that bind to cell receptors which cause a person to be infected. Like other respiratory viruses, SARS-CoV-2 spreads through droplets that are released when an infected person coughs or sneezes. These droplets can travel for 2 meters, so they can easily be transmitted to people breathing in those droplets. The droplets can also land on objects and survive between several hours or even days. A person can also be infected by touching his or her face after touching those objects. To protect yourself and slow down the transmission, clean your hands frequently with hand sanitizer or soap and water for at least 20 seconds in order to break apart the virus. Cover your sneeze into your elbow or use masks to prevent the transmission. So whenever you're going outside, use a 3-layered fabric mask correctly to ensure its effectiveness. But we have to keep this in mind. Masks are not replacement for physical distancing, hand hygiene, or other public health measures. So do yourself a favor by staying at home. Why? Because the incubation period for the virus itself is from 2 to 14 days. This means you could be infected and show no symptoms between this period. If you're practicing physical distancing, you would break the chain of infecting others and slow down the transmission. Keep yourself healthy by eating fresh food, doing some exercise, sleep properly, and continue to follow respiratory hygiene practices. If you experience any symptoms, isolate yourself from others for 2 weeks. If you continue to have symptoms, seek medical attention or call COVID-19 hotline provided by the government. This isn't just about medical workers or police anymore. We have to unite and fight this hand in hand to overcome this pandemic situation. Stay at home and be hero for others.
Hundreds of years ago, the world had been hit by several pandemics. Some of the worst pandemics recorded in history are the Justinian's plague, Black Death, and Spanish flu. Now, a new pandemic has been discovered in Wuhan, China. More than 6 million cases and 300,000 deaths have been reported from all over the world. It's called COVID-19. COVID-19 has been reported for the causative agent of pneumonia. Furthermore, this virus is identified as a member of the coronaviruses group called SARS-CoV-2. What is coronaviruses? Coronaviruses are a family of RNA viruses. They are called coronaviruses because the appearance of its spike protein resembles a crown. There are several of them that have been made an outbreak, such as MERS-CoV, which is transmitted from the dromedary camels to humans. What about SARS-CoV-2? SARS-CoV-2 is transmitted from human to human by the respiratory droplets when we are talking, coughing, and sneezing. The transmission from human to animal considered to be low as well as animal to human, and the research is still going on. Although genetically closely related viruses have been isolated from rhinolopus bats, the exact route of transmission hasn't been established yet. Now, the question is, how about COVID-19 infections in animals? Some animals have high susceptibility, such as cats, ferrets, and tigers, low susceptibility like dogs, and not susceptible like pig and some poultry animals. According to OIE, cases of animal infections have been reported by several countries such as mink farms in Netherlands, cat, lion, and tiger in USA, dog in China, and cat in some countries. The information about the transmission route is still limited. However, as for the other respiratory viruses, it appears that the virus is transmitted from direct contact like droplets. How can we make contact with animals in safe ways? First, don't let animals interact with other people or other animals outside your home. If a person inside your home is sick, isolate he or she from other people including animals. Always wash your hands with soap and water for at least 20 seconds before and after contact with animals. What should I do if my pet or animals are showing COVID-19 symptoms? Do not take your pet or animals to the veterinary clinic by yourselves. Please kindly call your vet. Your vet can then evaluate your pet or animals and determine the next steps for the treatment and care by phone or seeing them by themselves.
Thank you, Noli, for the video. Good afternoon, everyone. Good afternoon for the speakers. Thank you very much for coming. And maybe good evening or good morning, depending on wherever you are. Thank you very much for joining our international webinar titled With Pets as Zoonotic Pathogen Reservoir. My name is Putu Tri Devisca. I will be the MC for today's webinar. In this week, OHCC UGM will hold 2 international webinars about research update of wildlife-borne diseases. Today is the first session, and the following webinar about novel technology for wildlife pathogen identification will be on this Wednesday, on 9 December 2020. As you already know that wildlife has been strongly associated with the emergence of zoonosis and emerging infectious diseases, this fact should be a trigger for us as a researcher, the policymakers, and clinicians to make a step forward in regards tackling this complex situation. Through this webinar, we would like to update our knowledge and information about wildlife research, especially in pathogen investigation, what technology used in today's global research, what is the today's trends of research, so that we can fasten our research, making collaboration in very short time ahead. It is not in the future time anymore, I hope. Therefore, today, let me share my screen first. We already invite our outstanding speakers. Okay, I hope all of you already see my screen. We have 3 outstanding speakers. First is Dr. Joko Pamungkas, D.V.M. He is a lecturer from IPB University. He will talk about biodiversity in Indonesia and the potential disease transmission. The second topic will be presented by Dr. Jose A. Escarlos, D.V.M. Ph.D. He comes from College of Veterinary Medicine, Central Mindanao University, Philippines. He will talk about zoonotic disease transmitted by bats in Philippines. The last speaker is Mr. Serge Morand, Ph.D. He comes from Faculty Veterinary Technology, Kasetsart University, Thailand, Directeur de Recherche, CNRS-CIRAD. He will talk about the immunogenetics in wildlife. Every speaker will talk for 25 minutes, and we will have a Q&A session for 30 minutes as the last session. So I will not talk anymore. Before we start our webinar today, let me introduce you about the housekeeping items for all of the participants should be noted. First, all of the participants may ask the speakers during the webinar through the Pigeonhole link. through this link. I will share the link on the chat box, so you can simply click the link and write down your question there. And please tag the speaker's name that you want to ask for. Second is the other issue may be asked in the chat room. Your attendance will be only confirmed by filling the online form that's shared at the end of this webinar. And the e-certificate will be shared according to the attendance form and emailed about 1 week after the webinar. And the materials will be shared with the permission of the speakers. So I would like to invite Professor Dr. Nwayar Tunas Artama D.V.M. to be our moderator today. He is the coordinator of One Health Collaborating Center, Universitas Gadjah Mada, UHCC UGM, and he is also a professor from Faculty of Veterinary Medicine, Universitas Gadjah Mada. I will introduce you a little bit about his background. Prof. Wayan graduated from Faculty of Veterinary Medicine, Universitas Gadjah Mada, and finished his doctoral degree at the Institute for Veterinary Biochemie, Freie Universität Berlin, Germany, in 1989. Currently, he is a permanent lecturer at the Faculty of Veterinary Medicine, Gadjah Mada, Universitas Gadjah Mada. He was the Vice Dean for Academic Affairs, Research and Community Service at the Faculty of Veterinary Medicine, UGM, and got Research Award from the British Council. And he is actively conducting research and publication on toxoplasmosis, tuberculosis, and other zoonotic diseases, especially in wildlife. He has appeared in several national and international seminars. Now, Prof. Wayan is the coordinator of One Health Collaborating Center, Universitas Gadjah Mada, and an expert member of the COVID-19 Rapid Response Task Force Indonesia. I will invite Prof. Wayan. The screen is yours. Thank you. Prof. Wayan, can you unmute yourself? We couldn't hear your voice.
Okay, Putu, thank you so much for the wide introduction, short introduction. Thank you, everybody. Good afternoon. Selamat siang. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera untuk kita semua. Good afternoon, everybody from around over the countries and also from abroad. Today we have 3 speakers that is already introduced by MC. The number 1 is Dr. Joko Pamungkas from IPB. He is already well famous in the wildlife expert. I can introduce him in the CP for short CP. Could you please share? Joko Pamungkas, D.V.M, MEC, is senior researcher at the Study Center for Primate LPPM Indonesia, Department of Microbiology, Faculty of Veterinary Medicine, IPB Indonesia. He is lecturer at the University of IPB, senior researchers, Study Center in the Primate Center. Education, he got D.V.M at the Faculty of Veterinary Medicine, IPB University Indonesia, Master at School of Public Health and Community Medicine at the Washington, Seattle, USA, doctoral program in biomedical science at the University of Indonesia. So organization, he is a chairman of the Indonesian Biorisk Association since 2021, deputy chairman of the Indonesian Veterinary Microbiology Association, coordinator of One Health Laboratory Network in the Hun, start 2018 until now. He has published a lot of publication in the wildlife. So this is a short CP of Dr. Joko Pamungkas. Welcome, Pak Joko. Thank you for join the third time join our webinar. The second speaker. Okay, maybe because of the limiting time, you can share your talk at this time for use for 45 minutes, and then as a first speaker for this webinar. Pak Joko, time is yours. Okay.
Thank you, Prof. Mayan. Good afternoon, everyone, or good morning, good evening, anywhere you are at the moment. So as been introduced by Prof. Mayan, I am from the Faculty of Veterinary Medicine in Bogor Agricultural University, IPB, in Bogor. And I should consider myself not as an expert in wildlife. I have to study more about wildlife, yeah. But I happen to do some research on the viruses, surveillance of viruses in wildlife, especially in non-human primates and bats as well as rodents. So allow me to start my presentation. Okay, let me make it full screen. So I modified a little bit the title again because I'm not considered myself as an expert in bats, especially. So instead of having this bats diversity in Indonesia, I will not cover about that so much because I'm just doing some little works in bats, and I made some modification of the title, becomes bats in Indonesia and the potential of emerging infectious diseases or zoonosis in Indonesia. So my first slide, I have to put some disclaimer here. Sorry. This following presentation are based in part of my activities, our activities, and information gathered from published articles, but may not include all information available as of today. And contributors for this presentation actually are, among others, we have colleagues from the Primate Research Centers at IPB University, Dr. Tia Hiskandriati, Dr. Suryo Saputro, and also Dr. Uus Saipullah. And from EcoHealth Alliance, our counterpart from the United States, we have Dr. Kevin Olivo, Dr. William B. Karas, and also Dr. Alice Latin to be our main partners during the activities in Indonesia. So talking about wildlife and emerging infectious diseases, we probably learned a lot. And in about 2008, around 2008, there was a paper by Kate Jones, and stating from that article, actually, there has been some information around 75% of emerging infectious diseases in humans are zoonotic diseases. And among these, the zoonotic diseases, about 60% or more of these are associated with wildlife. So basically, wildlife has quite a role in the emergence of emerging infectious disease and also zoonosis. And I believe that you also have learned about the drivers of this emergence of EIDs. We could break into 4 categories: the environmental, human behavioral, industrial changes, as well as transportation. So basically, we can see here, if we put into percentage, the majority of the cause or the drivers of this disease emergence are basically land use change. It implies about 31% among the others. And this also has consequence in the deforestation, agricultural industrial change, habitat destruction or degradation, as well as habitat fragmentation for the wildlife. Yeah. And then others than the land use change, we also see here some human behavioral that may also have some influence in the emergence of infectious disease. Among those, the bushmeat. Yeah? So the practice of bushmeats in many countries in Asia, not only Indonesia, but also maybe some other countries, as well as in Africa. Yeah. Also, we noticed some agricultural industry changes here, about 15%, and also some other factors that might drive to the emergence of infectious diseases. The paper from Murray and Tesak in 2013 in Current Opinion Virology, this land use change could lead to the disease emergence by two theories or two hypotheses. The first is the pathogen pool hypothesis, and the second is the perturbation hypothesis. If we look about the viruses availability or viral richness in mammalian animal, we have some data from Kevin Olival 2017 published in Nature that some orders of animals actually consist or they harbor some viruses that might be zoonotic or might not be zoonotic. So among this viral richness in the orders of animals, mammalian animals, some are causing zoonotic diseases, and among other orders, there are 3 orders actually that are more contribute to the disease occurring in human beings, which is Chiroptera, the bats, the bats family. There, primates, non-human primates, of course, and also rodents, So in these 3 taxa, they have more viruses that might be able to be transmitted to human beings and cause some zoonotic diseases. Among the other orders, they have also some viruses, but they might, the proportion of the zoonotic cases are quite low. Okay, so zoonotic disease originated from bats. Let me move this block my view. Okay. Zoonotic diseases originated from bats. Among these emerging infectious diseases, I colored those that are in red, like Ebola and Marburg viruses, and then Hendra and Nipah viruses, SARS, MERS, and also the SARS-CoV-2 viruses. So basically, we understand that there has been some outbreaks, if not pandemic, for several cases, several diseases, like Ebola emerged in 1976-79 in Sudan and Zaire. Zaire is now called as Republic Democratic of Congo. And again, they emerged in 1979. And Marburg actually emerged earlier in 1960 something. Basically, it is transmitted to one of the laboratory technicians in Marburg, Germany, because of the contamination of kidney cell culture that they brought from Africa for some research in the lab. And then Hendra and Nipah virus, now they are grouped into one genus, which is a Nipah virus. Hendra originated from Australia, and they actually noticed that the occurrence of this disease were in horses. And the first veterinarian who treated this animal because of the no information about this pathogens before, so he didn't put quite proper PPE. So then the first victim in human beings of Hendra infection is actually veterinarians. That was in 1994. And then 4 years later in Malaysia, there were also an outbreak of this encephalitis diseases in farmers. And it was also associated with some pigs as an intermediate host. So basically, they discovered that the Nipah virus has close relationship in the genetic level with Hendra virus. And Nipah not stopped in 1998 and 1999, but it becomes also spread around into several countries in Central Asia, like Bangladesh and India. And And the speakers from Malaysia, from Philippines, today we'll also talk about this Nipah virus in Philippines. So we will learn about that. The SARS and MERS also emerged in 2002, 2003 for the SARS, and MERS emerged in 2012 to 2013. These are caused by beta coronaviruses, and the study has shown also that there are some closely relatedness of these viruses that cause SARS and MERS in human beings. Yeah. And of course, today, nowadays, we experiencing this pandemic of COVID-19 that is also caused by beta coronaviruses. So this is also something that I wanted to share with you about the viral transmission from wildlife. We learned that there are two patterns actually, that wildlife as a reservoir, they harbor the virus without showing any clinical symptoms. And one pattern, they could actually directly transmit the pathogens onto humans, like the viral pathogens, in this case, like Nipah, yeah, Nipah virus, as well as the rabies virus. They could be transmitted directly to human from this reservoir. In this case, we understand that this virus is actually at the stage to be ready in infecting human beings. So they don't need intermediate hosts. The second pattern, this pathogen will need intermediate hosts, and it could be the wildlife, yeah, animals, as well as livestock. Yeah. So in the case of Nipah, we learned that for the direct transmission might be occurring in Bangladesh, where they don't have like an industrial farm for pig. But in Malaysia, we learned that there was an involvement of these intermediate animals, especially pigs, and infecting the farmers. Yeah. For the coronavirus, merely there was no report about direct transmission from reservoir to human beings, but they always need an intermediate intermediate host to infect humans after some evolution of the virus in these animals. Yeah. These are the intermediate hosts of coronavirus, the beta coronavirus that has been reported in humans. The first one is SARS-CoV in 2002, 2003. These animals has been known as intermediate animals, like the CFATs and also some other carnivore animals. Yeah. And then in 2012 and 13, reports also showing that the camels, yeah, so especially from the Saudi Arabia, yeah, causing functioning as a role of intermediate animals before infecting to human. And the current pandemic, we also learned that there was some closely relatedness between the bats SARS-related coronavirus in Chinese horseshoe bat with the one that already published from the humans in China. Yeah. And still some questionable about which animal are actually functioning as intermediate host. Yeah. So challenges in studying viral infection in bats. We basically are not quite, it is not quite a normal procedure to do surveillance of viruses in bats. So we need to actually continue and do this periodically. Yeah. So throughout the years, we should conduct surveillance of viruses in bats and other wildlife animals. And also the challenge is in the detection versus the diagnostic laboratory tools. If we are applying the diagnostic laboratory tools, which is quite specific for certain viruses, we may not be able to catch something different that has not been reported. So basically, we have to use the detection, or what we call it is a discovery tools, yeah, enable to detecting the virus, which is probably not reported previously. And we should also conduct some concurrent surveillance of viruses in bats, humans, and also livestock. Okay, so in the case of the emergence of infectious disease, this is a graph that Dr. Karas actually published in 2012 in, I believe it is in the Lancet, yeah, the Lancet, yeah. This is something that occurs normal in the nature, that basically we or several cases, several outbreaks are noted in the human beings. Basically, they are the spillover from wildlife. So if we continue in doing the surveillance of viruses in wildlife, we may be able to have some patterns of the map or the distribution of the virus in the nature. So by having this information, we could actually make some alarm or warning systems to the human, also to the livestock. Yeah. I mean, to the authority for the human health and the authority of the livestock. Yeah. So if we have the information about what viruses are circulating in the nature, so basically we could make an alarm to these two sectors of health. Yeah. Okay, this is just to give you brief information that USAID sponsored a program called PREDICT. This is one of the USAID's emerging pandemic threats program. And it is a global programs or projects that are run by the consortiums in the United States, consisting of 5 organizations led by the University of California, Davis. And this project actually is using the One Health approach to understand the dynamics of zoonotic virus evolution, spillover from animals to people, amplification, and also spread to inform prevention and control. So talking about the practice in Indonesia, especially, I mean, again, this is not specific or unique to Indonesia, but also happening in other countries. We have some activities of wildlife trade. Yeah. So bats as one of the wildlife favorite in certain areas in Indonesia, especially in the North Sulawesi. Yeah. So they actually have several types of habitats in the caves or maybe in the neighborhood area of the community, as well as in the mangrove forest, yeah, mangrove. And they will be hunted and be kept for certain times at the pool sites, yeah, before they are picked by the middleman to be transferred to the markets in North Sulawesi. Yeah. So this is the situations that we normally see in the wet market, or we call it sometime, what do you, how can I forget it? Okay, but anyway, this is the picture that we took in several markets in North Sulawesi. And not only bats and rodents, we could also found that they actually sell wild non-human primates that are actually endangered species. So this is about the trade sampling in Indonesia. So there has been unregulated hunting and trade threatens to 2 species, especially Azarodon species and also Pteropus species. Why these 2 species? Because their size are quite big compared to the others, and these are having a lot of meats, and they like to consume this because of these 2 reasons. Yeah. So wildlife trade in Indonesia basically occurring mainly in North Sulawesi. So Sulawesi has 6 provinces: North Sulawesi, Gorontalo, Central Sulawesi, West Sulawesi, South Sulawesi, and Southeast Sulawesi. So in North Sulawesi, especially with the community of Christian people who likes to actually eat or consume these bats and rodents as well as non-human primates for certain occasions. Yeah. And this has been very intensive trade in the regions, and we identified at least 8 main wildlife markets in these regions of Minahasa especially. Yeah. So this map actually is about what we try to capture, and also we try to see some activities in the markets. Yeah. So when we further studied that the source of the bats or the animals actually are not from North Sulawesi itself, it may be because they have exploited in the past and they have consumed a lot from their area. So then it become extinct and they have to import it from different provinces, from West Sulawesi, from Southeast Sulawesi, Central Sulawesi, as well as from Gorontalo provinces to be sent to North Sulawesi. Yeah. What we found of our first stage of study in detecting some viruses, I forgot to mention that the predict actually has 2 aspects of surveillance in human and also in wildlife. So the human surveillance is conducted by our colleague from the Ekman Institute for molecular biology, yeah, and then we conducted the surveillance in wildlife. So from the first stage, from 2011 to 2014, we found several viruses. We detected several viruses, such as paramyxoviruses, coronaviruses, astroviruses, rhabdoviruses, as well as herpes viruses. So this is using the predict protocol that actually we applied some non-specific PCR. Basically, it's a broad spectrum PCR that can detect members of genus as well as the family of viruses. So not in the species level, but in the genus or in the family level. Yeah. So talking about this, we also have some information about the distribution of bats that has the SARS-related coronaviruses in Southeast Asia. But if we look further, that in Indonesia, we also have some species of Rhinolopus avenis, yeah, distributed in the islands of Sumatra, Java, also several areas in Kalimantan or Borneo, as well as in Lesser Sunda, yeah, Nusa Tenggara. So as a conclusion or maybe some closing, we have some submitted some policy option and economic tools for common pool resources. Basically, we cannot stop right away about the practice of bat hunting, but we could develop the association of bat hunters to increase price, yeah. So then it will reduce the total consumption and also develop sustainable hunting. So if we are to stop right away, it may not be happening. So we have to do it step by steps. Also, we suggest to the authority to control for the bans for several seasons. Yeah, so short seasons drive up cost and protect the bat stock. So meaning that during the short seasons, the price will increase, and then probably people will not buy that. And by doing some interview, we noted that we could actually do several steps in order to stop these practices. Last but not least, we also notice overhunting bats generates societal problems. Yeah, in the public health area, there are increased risk of virus spillover driven by the human-bat context via hunting, slaughtering, and trading behaviors. And we noticed that there are some paramyxoviruses, the genus where the Nipah virus and Hendra virus also there. And also we noted some coronaviruses, especially the beta coronaviruses. So in the conservation and sustainability manner, the declines in abundance and diversity of bats, losing important ecosystem services, while unsustainable livelihood from the value chain among hunters, middlemen, market traders. So we identified that the high risk community actually are not the consumer, because they are not touching the bats in the live condition. They just came to the restaurant, the bats already been cooked, well cooked, so they never has any interactions with the wildlife while they're alive. But the hunters, middlemen, and also market vendors that probably prepare also for the restaurants, they are the high risk people. So this is what I could share. So thank you very much. I would return the time to Professor Wayan. Thank you very much.
Thank you, Pak Joko. Very nice presentation. I think this 45 minutes is not enough to hear what you have already experienced. Pak Joko concerned about many things. First, about the drivers of the infectious emerging infectious disease, including climate change, agriculture change, industrialization, and transportation and globalization. And the second, he mentioned also observe the richness, viral richness in the mammal. And then the third, zoonotic disease originated from the bat, like Spanish flu, Ebola, HIV, Hendra virus, Nipah, Zika, influenza, and COVID-19. And the last one, he also mentioned about the virus transmission from wildlife to the human being through livestock, wildlife as an intermediate host from the bat. And then last one, he also mentioned about the livestock trade in Indonesia and challenge to studying viral infection. There are many viral diseases already identified in Indonesian area, like parvovirus, paramyxovirus, coronavirus, astrovirus, rhabdovirus, and herpes virus. This is time for you, for the participant. You want to ask a question to Pak Joko, please ask in the pigeonhole. And our second speaker, thank you, Pak Joko. And our second speaker is our guest from the Philippines. He is Jose Escarlos from the College of Veterinary Medicine, Central Mindanao University and in the Philippines. He will introduce we learn from lesson learned from the Philippines, the transmission of the disease through bat in the Philippines. I think this Jose Carlos is a PhD and he's got a PhD and is a professor in the Central Mindanao University in the south of the Philippines. He has an administrative assignment in the university as a director of the alumni relation and linkages, and currently active with the research on biodiversity in Mindanao, especially on the bat, with the Center of Biodiversity Research Extension in Mindanao. He also collaborates with the GRACE Network, also with us in Indonesia. And I think it's time for you, Josh. It's time 45 minutes for you. Okay, Josh, it's time for you. Hello, Josh, you can share the screen. So what's happening with him?
Jose, ini Prof, jadi attendees mungkin dimasukkan jadi panelis lagi.
So Jose is already in panelist.
No, now he is in the attendees. Maybe Putu.
I think he was out for a while and joined to be an attendee, and I already put him as the panelist. Nah, here you are. Dr. Jose, the time is 25 minutes. Thank you very much.
Yeah, I hope I could do the justice in.
Okay. Okay, please, Josh.
So good afternoon to everyone. Can you hear me?
Yeah, very clear.
Okay, greetings from the Philippines. I am Dr. Jose Escarlos Jr., and I hope you are all comfortable and you can hear me clearly. I am given the task to present our paper together with Dr. Alan Dargantes and Dr. Mohagan, who is a biologist. Dr. Dargantes is an epidemiologist. I told him to come here and we present together, but he is so busy because he's in charge of the bidding activity of the university for our supplies and materials. So I was part of the team of Professor Artama, the one which we have discussed a while ago, Professor, the one in Komodo Island. And Dr. Artama, I would like to thank you for that wonderful experience in Indonesia. Indonesia indeed is a very beautiful paradise.
Yeah.
Let me start my topic by introducing to you my country.
Josh, I just get mentioned from the MC, 25 minutes for you, right?
Yeah.
Yeah, okay, you can manage that.
Archipelago consisting of more than 7,000 islands home to 100 bat species, the Philippines is considered to be a biodiversity hotspot of high species richness and endemism. That's according to Mayors in 2004.
Excuse me, sorry, Dr. Jose, we cannot see your presentation.
You wait.
Yeah, you should hit share screen, please.
Yeah.
Wait, this one. Share screen, can
you can click the share screen, the green icon at the bottom of your screen.
Yeah, you wait.
Okay.
Share screen. Yeah, okay.
It's
lost.
Coming.
Yeah.
Is there?
Okay, fine.
Okay. Can you see me now?
Yeah. Okay, thank you.
Thank you. Yeah. Okay, again, I introduce to you the Philippines. It's an archipelago like Indonesia. It's composed of 7,000 islands and home to 100 bat species. The Philippines is considered to be a biodiversity hotspot of high species diversity, where it is known of a lot of things, like it is high in human population, a lot of forest resources, but overutilized. Now, my group encountered 17 species of bats, and 29% are threatened one. And the famous flying fox and the bald-backed bat is endangered. This is the result of too much resource utilization. My involvement in bat research is with the University Center for Biodiversity Research and Extension in Mindanao. Our center collects data from permanent plots established in 5 long-term ecological research sites in Mindanao. With this, another team is doing the same research in Visayas and Luzon. We report our data in the National Elter Forum to produce the Philippine data for our Southeast Asia counterparts, and Indonesia is a member of it.
Dr. Jose, I'm sorry, we see your note, your slideshow is screening your note.
Ah,
It's not in the full slideshow. Is that okay for
okay,
you?
I'll go back. Oh my God.
Maybe you can hide the presenter view or adjust it.
Okay,
First.
I'll go. I'll exit. Okay. Tabangi ko be. Tabangi ko clip. Nawala na sa naman niya. Ibalik ko. Makit-an dito nila ako. Ha?
I think in the bottom you have the like screen icon for full screen.
Oh, there. Okay.
Yes.
So
Yeah,
my
that's
task.
perfect.
Okay, my task is to assess status of bats, whether the species are critically endangered, vulnerable, or endemic, economic, etc. We classify them also and determine the biodiversity indices based on the establishment of priority areas for conservation, and we enhance bat biodiversity awareness and capacity building among communities. So this is my team. So my team goes to the mountain, and you will take note that this is an old picture. We were not yet wearing our PPEs. And mind you, today we cannot do this one. We never have went back to the field. And next time when we go back, we'll be wearing already our PPEs. We were just protecting our hands. So we do mist knitting, morphometrics, tagging, feeding, and then we release the animals to the wild. Then we process our data and computing diversity indices and the IUCN read list, assessing of the status of our animals. These are sample results of our Marilog district. This is in southern Philippines. These are data that we have in one testing site. And we do, we summarize them as the conservation, ecological status, and then the diversity, we compute them using the Shannon and Weiner. And then we also document these bats. And we also, part of our activity is to train local researchers and our students at Central Mindanao University. And we have made, we have advised our students to, in their dissertation, masteral, and their thesis work. Now, this is a sample of the summary of what we are doing. In just one mountain, there are 13 faunal species, and all other informations are fed to the center, to our center. Now, part of our activity is also to produce information, is the information dissemination that we do. So together with other researchers in the center, we produce flyers to disseminate information and to train people how to protect wildlife. Now, actually, our efforts on bats was started or pioneered by Ingle and Henley. Together, they made a comprehensive assessment of bats in the Philippines and developed the first 35 taxonomic key, which become fundamental to most studies in the country. So what do we have in all the studies that were collated? The Philippines hosts 80 plus bat species belonging to the family, to the 7 families. Subsequent discoveries and taxonomic revisions by authors and colleagues have increased these numbers to 103 and 25 additional species with description of bats in the Philippine Islands. We are discovering more. Approximately 32% of the bat species in the Philippines are pongiferous, or fruit eaters or nectar drinkers, and 37 remainder are insectivores. Further, For example, the flying fox, for example, because of high proportion in the Philippines, bats rely primarily on the forest, and the flying fox, for example, selectively roast and forage in the secondary forest, and even getting closer to man. While around 30 species roast in caves and underground habitats. There is the intensification of agriculture and other land use changes here, and I think our bats are having a difficult time. There are also other wildlife in the Philippines. Only in the Philippines, we have the tamaraw. I think you have a version there in Indonesia. We have the Palawan birded pig. We have our Philippine brown deer. I think this is also common in Indonesia. Then the bats and the Philippine pangolin in Palawan. And you will notice that most of our animals are quite smaller compared to other Asian counterparts. But we have the second largest eagle in the world, our national bird, and it is endangered, and there are only 400 left, and most of them are found already in the conservation area in Davao City, the Philippine eagle. So we have a summary of what we know. This is an old data in 2015. And of course, our main topic is about zoonosis. What is zoonosis? It is a disease which can be transmitted to humans from wild animals. And this is going viral this time, and it's the bat that is our topic. So we also have the definition of emerging diseases, which was well discussed by the previous speaker a while ago. And this is defined as infections that have newly appeared in the population, have existed previously, but are rapidly increasing in incidence or geographic range. So these are what we had experienced in the Philippines: nCoV, rabies, Nipah virus, and so on. First, let me tackle about rabies. Briefly, rabies is an RNA virus. It's very fatal. And about 258 human deaths in the Philippines. It is one of the Philippines is one of the top 10 countries of highest rabies cases in 1918. And because of this, the Philippines made the Anti-Rabies Act of 2007, implemented it. And the target is to make the Philippines 2022 rabies-free. So this is the appearance, the illustration of the rabies virus. And the carrier of this one are the stray dogs, and they are the primary sources of rabies in the Philippines. But originally, we learned that they came from wildlife, the rabies. It could be the civet cat, the monkeys, or probably another wildlife from the bats. And infecting the dogs or farm animals or human population. But in the Philippines, it is focused on this one, from dogs to humans and dogs to farm animals. So, of course, we have learned this in our pathology, the rabies pathogenesis. Okay, it is transmitted by bite, and that is how it's being transmitted in the Philippines. And incubation would be as early as 7 days, less than 7 days, or it would be as late as 6 years. And the average is 4 to 6 weeks. So we have stages for this one. And what is happening is almost all affected ones are dead. There were reported to have recovered, but the recovery was still on the prodromal phase. Now, we diagnose rabies by illustrating negro bodies or your fluorescent antibody technique, the cytoplasmic localization. And then we do prophylaxis in the Philippines, pre-exposure vaccination upon request, or you can spend your own money. And of course, post-exposure prophylaxis is for free in the Philippines. So rabies vaccines for dogs and rabies immunoglobulins in the Philippines. for human beings. So we have some guidelines how to prevent or treat rabies. And then our university is highly involved with rabies awareness. Our students are sent to lecture in elementary schools and high schools. And then, of course, with this very high incidence, we have 280,000 bites per year recorded in our Department of Health. Now, we are also threatened because we're so ashamed that a Norwegian woman died of rabies in the Philippines. So it threatens our tourism, and also this means a lot of money because we have plenty of cases and the government will spend so much for this one. Okay. More on the dogs than the cats. And to cut the transmission, we have established immunization for the dogs and the dog population. And of course, we increase public awareness in mass media. So to summarize everything for rabies, these are the things. We have vaccination for animals, vaccination for humans sometimes, and surveillance study were done, social mobilization, a lot of research, local program implementation, and information dissemination. The next infection that I'm going to discuss is the one very close to my place here in the southern Philippines, the Henipa virus infection. These are two zoonotic members, Hendra virus that occurred in Australia and Nipah in Malaysia. I think this was well discussed a while ago. The reservoir hosts are your fruit bats, but no illness has been observed in the fruit bats. They account this that to the resistance of the animal, that it increases its temperature during flight. Now, this is the extent where Henipa virus has affected. It occurred in these areas. In 2014 in the Philippines, signs were observed in horses, neurologic signs such as head tilting, circling, ataxia, rapid progression, and of course, death. Then we also have signs in cats, bleeding in the mouth and nose, and there were 3 deaths. These cats consumed the meat of the horses. So this is the possible transmission routes of Henipa virus that happened in the Philippines. So the bat droppings, it could be probably the fruit, or which was licked by the bat, or probably the droppings of the bat that contaminated the grass. Goes to the horse, and of course the horse was butchered or eaten by man, because in the place where it happened in southern part of Mindanao, the people there are fond of eating horses. So the sick person was brought to the hospital, the nearest hospital, and then the one taking care of the person were able to touch the secretion, and the health workers became sick also. That was the case of possible transmission routes of Henipa virus in Ninoy Aquino, in the southern part of Mindanao. So geographical distribution of Nipah is shown in blue. Okay, the next is avian influenza, which is the highly pathogenic avian influenza. This outbreak happened just recently, 2017, and the possible source are the migratory birds. And the ones affected here were the quails and the chicken, and this was contained, and we are now free again from the highly pathogenic avian influenza, but we're still under threat. So this is the distribution in Southeast Asia where your virus was observed. Okay. For human cases, okay. So this was in August 2007. The government was very prompt. From August 3 to August 23, we were able to check the problem. There was mass destruction, 1 kilometer zone circumference, and we butchered 37,000 birds immediately. An outbreak in Jaén and San Isidro New Barcija on quails. Again, this was done again. And then we killed 603,000 birds, ducks, and game fowls, and 115,000 quails. And the outbreak was declared clear on the 23rd of August 2017. So public information, control of bird movement, sacrifice chicken, ducks, quails, and affected areas, disinfection, concerted effort, and this was the solution to contain the HPEI. There was no human clinical case, luckily. Next is the Ebola restone. Ebola restone was first identified among monkeys in the Philippines in 1989. These monkeys were exported to other countries because they use this for the laboratory. Ebola restone is often fatal in monkeys, and 4 known cases in human eliciting immune response, but they are asymptomatic. So Italy and in US outbreaks on monkeys from the Philippines. But what was affected in the Philippines are the pig population. First outbreak of Philippines is in 2007 and 2008. We account it as the PRRS and the Ebola restone. So the first pig-to-human transmission probably was from the wild pigs. Detected 6% of tested farm abattoir workers, no, and asymptomatic. They are also asymptomatic. We have done our research on butchers, and thousands of pigs were sacrificed also. And I think we are containing the virus. So this is the extent of Ebola restone in monkeys and pigs infection in the globe. So possible transmission from bats to bats, or from bat to the monkey, or from droppings, and then to human beings, and probably human to human transmission. So what about coronavirus disease? Worldwide, last night I downloaded this one. We have now 66.6 million infected all over the world cases. We have 1.5 million deaths. In the Philippines, we have 439,834, and we're very worried this time. This time we don't have face-to-face contact with our students. I'm just here in my office with my assistant, and nobody is around the campus this time. There are other arbovirus which was transmitted by a mosquito: dengue, Japanese encephalitis, Zika, and chikungunya viruses that is experienced right here in the Philippines. So it's the female that bites usually, and they're engorged with blood. So this is the possible transmission, a general transmission of arbovirus. So the virus is transmitted by the vector through the vertebrate host. These are mammalian, usually mammalian species, and then bitten again by this one is bitten again by another non-infected vector, and that the transmission goes on. So there are This is highly prevalent. Dengue is very prevalent in the Philippines. A lot of children are dying. And what we have as a treatment in the Philippines is more of fluid replacement, and the doctors here are very good already in treating dengue, infusing blood and maintaining the level in such a way that the human being is able to resist the disease. And we have vaccination, but it got a controversy in the Philippines. And as of the moment, it's not yet accepted in the Philippines. So these are the countries with most cases: Brazil, Mexico, Nicaragua, Philippines, and Malaysia. So Indonesia is not included, I guess. Now, the Philippine dengue cases in this December 14, 2019 was this much. In December 2018, it was this much. And finally, our new record in the Philippines, it goes down by 66%. I don't know if we're improving in the cleaning our surroundings and making this mosquito to be not so abundant in our locality. So I think we know this. Then we have Japanese encephalitis, signs of high fever, chills, headache, fatigue, and death. This was confined for quite a time in Luzon from 1958 to 2013. So the same transmission, mosquito to animals. And our group here in Central Mindanao University studied the dynamics of the transmission of dengue. We detected pigs and buffaloes, cattle, goats, dogs, and rats. This is a research of Dr. Baltasar, my colleague in the university. And then there are a lot of studies since 1966 to 2005, but for me it's scanty. And then we have the 2015 study reporting your GEV. Then we have other emerging diseases, the Japanese encephalitis virus, isolates, okay? And they were able to show that they are related to each other.
Sorry,
So,
just 5 minutes left.
Oh, okay. Yeah. So we have also Zika, and it is confirmed in the Philippines, chikungunya, and of course, our friend the bat is the main source of many diseases, and he licks our fruits, do his droppings, and then go near to us because we have destroyed the forests. And then because of this, the population of the Philippines has increased so much, and some even eat these animals, poor animals, and then we have researches conducted on that. Okay, these are the red spot is more on the diseases research. Very few, it's more of biodiversity researches. So we have studies in bats. We have studies also on DNA barcoding of bats itself done by the UP Biology Department. And we have data done by Dr. Masangkay and his Japanese friends of the University of Japan and the University of the Philippines, detection of new bats gamma herpes virus in the Philippines, and our first molecular evidence for beta coronavirus in Mindanao was done here in Mindanao, together with collaboration in Japan and Australia. And we have a newcomer from Japan who worked on encephalomyocarditis, Dr. Carla Doisabas, who is my colleague here in the college. She will continue her research on virus research, and she will make collaboration with Dr. Wayan, I guess. And of course, the small choices we make each day can lead to the kind of world we all want for the future. Terima kasih untuk mendengarkan. I hope I pronounce it properly. Thank you for listening.
Thank you, Jose. Thanks so much for your presentation. And I don't know that Philippines have also a huge island, 7,100 islands.
I think you have more. You have more.
We have 17,000
Yeah.
islands. So
We are called as the
10,000 more.
before by the
But
American.
both of us, we have diversity on the bat and also on the many wildlife diversity. Jose have already explained us what is the emerging infectious disease in the Philippines. Like they have here, they have over there a rabies problem, Nipah virus in Hendran and Nipah HPI that we have also now. Until now, we still have HPI in Indonesia. And he also mentioned that Ebola reston in pig in the Philippines, but you never have African swine fever, right?
Yeah.
Arbovirus transmission and how to control dengue. You have very success in controlling dengue, but we also in Indonesia we using bacteria to control the dengue as a pilot project in Yogyakarta, Wolbachia. And we have also problem with Japanese encephalitis. I think this is what Jose have introduced to us as a lesson learned from the Philippines. And if you have any question to Jose, please write down in the pigeonhole, and then he will answer your question. And this, our next speakers, we have the third speaker, Serge Morand. He is now in Thailand, but originally is from Montpellier, France. So may I introduce him? Serge Morand focused on his research on the evolutionary ecology of disease transmission and applied health ecology. Field parasitologist, he is concerned by the interlinks between biodiversity and health, investigating zoonotic infectious disease, foodborne parasite, antimicrobial resistance in human at the human-animal environment interface. So he has conducted numerous field trips in Southeast Asia like Thailand, Cambodia, Laos, Vietnam, in the Philippines, and also in Indonesia. You didn't mention that.
Not field trip, not yet.
We have a collaboration research and lead several projects on the impact of land use and climate change in biodiversity and health Southeast Asia. Serge Morand belongs to the French CNRS-CIRAD, currently based in Thailand in the University of Kasetsart University, and also invited professor at the Faculty of Tropical Medicine, University, Medan. I think it's time for you. Okay, Serge, you can share your presentation.
Okay, so 25 minutes. So
yeah.
I put this.
Okay, thank you.
Okay, so I go to my presentation, partager l'écran. Okay, do you see my screen?
Yeah.
Okay, so first I would like to thank you and Professor Wayan to inviting me, and I want also to thank the two first speakers, Dr. Jose and Dr. Joko, because they are really putting the good background and more easy now to go and to discuss a little bit something that maybe I would like to do that not too complicated. Simplify on how to show that finally knowing parasite and viral diversity, and we can link that to immunology, immunogenetics of wildlife, and we maybe better, I would say, help to understand disease transmission and potentially risks spillover. And again, I'm very happy to give this talk because it reminds me that during the last more than 20 years, 25 years, I still continue to ask this question: why there are so many parasite species? Ectoparasite, endoparasite, helminth, protist, viruses, bacteria. Why so many? And this, I published several books and studies on this, knowing that 50% of the life are actually parasite of the other living. And on this, we can find the diversity, and I put only these figures to show that there is a differences between according to the big group of hosts. But mammals and birds, we already discussed, are very good hosts for many parasites, at least for the big parasite like the helminths, but maybe other. And within the mammals, I look at the diversity of ectoparasite, how many species of ectoparasite we can find on each members of mammals within the family. And we found that there is also huge variation with some groups of mammals with very low number of ectoparasite, but with a group that are very good to host ectoparasite, which are the bats, but with also the rodents and also all the group of shrews. Very good one, even if they are not related by phylogeny. And something starting quite interesting, when we look at the diversification of hosts, we found that according to the data, there is the ectoparasite diversity and maybe some other parasite, the diversity of parasite in general is really favoring the diversification of hosts, which means that it's not so astonishing that we found that bats and rodents are very diversified because they are also hosting a very diversified parasite and also viral communities. So my question was always like this. So how to explain this parasite diversity, how parasites adapt, how hosts adapt. So finally, what are the relationship between host and parasite? And there is asymmetry between host and parasite because, as we have already seen with the precedent presentation, a host, like for example, the human being, we are facing multiple parasites. We have many parasites that can infect us, at least more than 1,000, 1,500. We are one of the species that are hosting and that are suffering the high burden, which means that we have to cope with all these parasites. So what we have already learned that was important to explain the parasite diversity, if we look only at the helminth or the helminth, the protist, the metazoan, we found that mostly it's geographical range and host population density, which means when the host live in high density, large population size over a large geographical range, this may explain why this host is also infected by a highly diverse parasite communities. Actually, we will focus now mostly on bats, on rodents, and first with the viruses. Why? Because of the coronaviruses, for example, when we look at this paper of Zheng Lishi and published several years ago, not so long a time ago, and when she compared the origin of human coronaviruses, what she found in this is mostly bats, but also rodents that were the reservoirs of these coronaviruses that emerged to humans, but mostly by intermediate hosts, and mostly they are livestock, or recently wild mammals that are putting in farms, like the civet cat. And just to compare also, because when we look at bats and rodents, already said that we have around more than 6,000 species of mammals. Bats, 20% of the mammal species with 1,400 species. Rodent is a little bit more than 2,000 species, 30% of mammal species, which means altogether 50% are bats and rodents, and they are living differently. Of course, bats are flying and rodents are grooming on the ground, or going on the trees, or also in the houses. They are living in a different environmental niche, but they are close. They could be close to us. And we are also doing a lot of works, as we have seen before, with the virome of bats. But we are just finishing a paper also with our colleagues from China, but with some colleagues of EcoHealth. We already discussed about Alice Latin, so she's there, but also colleagues from Thailand, Cambodia, and Laos, and to describe all the virome from about 3,000 rodents all along the Mekong in Cambodia, Laos, and Thailand, and we found a huge diversity of viruses for more than 200 viral families. So with many species of viruses in DNA and rodents. The paper will just appear in a couple of days now, and we will do a little bit more to go there, and maybe to links with the immunogenetics. So with all of this, we know a little bit better about the diversity of DNA and RNA viruses, but also for many species of mammals, including the humans, rodents, and bats. And as you can see here, actually, apart from the primates, most of us, rodents with high diversity of RNA viruses and bats are really important in terms not only about the diversity of their viruses, but also about sharing this diversity with other species, and especially with the primates, but not only, but also with other mammal species. So quite important. What we know about to explain the bat viral diversity, I would say that we don't have enough information, not enough, and I will show you first what I did a review of this. And for the moment, colony size seems not to be important traits of the bat to explain their viral diversity. Not their ability to migrate, not their roosting, but fragmentation seems to be, habitat fragmentation seems to be important in their viral diversity. The same when we look at another, just to show before that there is maybe something else. We compare for the bats, but from South America, and we found that the brain of the bat, the size of the brains compared to the body weight seems to be important. It's related at something that finally investment in the brains, investments as a life trait, maybe explain that there is a decrease of investment in the immunity, which may explain why the bats are also a good reservoirs for parasites. So we looked at fragmentation, and we found effectively that fragmentation is really the most important thing with the host weight to explain the viral richness in African bats. When we look at the viral species richness in Southeast Asian bats, we found a little negative influence of colony size, but really negative, but mostly importance again of fragmentation. But what I have to say that look at the number of species that were investigated compared to the number of species we already knew that are present in Southeast Asia. We have two precedent speakers that have really focused on this. We have very few species investigated. And what we found here that most of the factors that explain the viral diversity is the sampling effort of the researcher, meaning that sampling efforts explain the viral diversity, which means that we need to sample more bats. We don't need enough information. So we hope that all the PREDICT project and other projects on the virome of bats will try to increase of knowledge of the virus diversity in bats, so we could better understand the factors that explain among bats their diversity. And there is another paper quite recently that tried to find the differences between bats and rodents at reservoirs of zoonotic viruses. In rodents, they found also that the rodents that live together that are important. On the bat, geographical distribution, so we already found that, but related also to the fragmentation of the habitat. They found body size, already we found this body size, but they found also the longevity of bats, which seems to be important, and also the birthing, the reproduction of the bats, which is also quite important in terms of immunity, because when bats reproduce, there is a decrease in the immune responses, the immunocapacity, there is immunomodulation that explains that there is an increase of the viremia. Much of the bats have higher viremia during the pregnancy or during the reproduction of the bats. But we have to say that, again, in this paper, they found all of these factors are marginal in terms of explanation compared to the citation count, which means that, as if we emphasized before, they also emphasize that we need additional data to be better in our comparative studies. So there are several papers that try to find why bats are good hosts for zoonotic viruses, maybe related to bat flies, maybe related to bat torpors, maybe related to the metabolism of bats. So several of these are, which is difficult to test, because this is bats for the flight is the only mammal that can fly, so it's very difficult to compare. But we should maybe compare, especially with the metabolic rate. I think we can have something on with the reproduction. So bats on immunogenetics related, and I will do a comparison with rodents. And again, I would like to thank Professor Wayan for inviting me, because it reminded me that I'm starting to do that. Wow, 15 years ago, but for fish. At this time, I was interested in fish, at the polymorphisms in the MHC, the immune genes in fish, and actually we found a lot of interesting stuff related to MHC variability, life threats, and parasite diversity for cyprinid fish in Europe. And that's why also we did that in rodents, actually, first in Europe, at the level of one species, and it was done with one of my former student, Joel, that now he has a high position in Czech Republic. And we did also a comparative stuff, and we found again that there is a correlation between the diversity and the genetic diversity at immune genes and the diversity of elements within species. We did that 5 years, 6 years ago with all the rodents we collected in Southeast Asia, and the same. So now we will do that with the virus, but at this time we look only at the elements, and we found quite a good correlation between the diversity of the immune genes, and especially the DRB alleles using NGS technology, and with the diversity of the elements. And we look like at the level of individual, but at the level of the species, showing that, okay, parasite maintains the diversification of immune genes, and they are very important in this. We look also at some other genes, but mostly in Europe, like the toll-like receptors that are more related to some other virus, some other parasites. Finally, what we know about bats. At the moment, not so much. Not many people were looking really at immunogenetics during the last, I would say, 15 years. It was not at the main host to be interesting, and especially in immunoecology. But now, of course, it's changing a lot. And I would like to share with you this paper that was published in Nature a couple of months ago. Very interesting, because they show that we can do now a full genome sequencing, and especially of the genes interesting on immunity in bats, and we can compare nicely the diversity the bats are doing, and also the genes that implied also in the immunoregulation on the ability, finally, to maintain on 2 months the immune system. But of course, for virus, we should not look only at these genes. We should look also at the proteomics, and especially all the non-coding RNAs, which are very important in a way, because these RNAs are important to cut the virus during the virus infection, on all of this diversity and all of these the number diversity of course of this expression of non-coding RNA is important to understand. These non-coding RNAs also vary according to the diversity of virus, but according also to the season, according to the species. So now we can, and I will conclude. Finally, I'm finishing a little bit more early than I expected, but we will have more questions now to investigate it. First, what we know that bats are virus-rich hosts, as well as other parasites. So bats are quite unique, maybe with rodents, in the number of virus, bacteria, and other parasites that can host. So this is quite important things to know. But some factors can explain this diversity, mostly related to some life history traits I discussed already, maybe the longevity, maybe the reproduction, but also related to the habitat and their geographical range, and maybe the habitat fragmentation that are important. So I would say that the understanding, the knowledge and diversity of parasites and viruses in bats and rodents are making important progress, but also thanks to the new molecular tools, the NGS, all these through output technologies. Now we can do microbiomes, virome, parasitome, protistome quite easily, and sometimes non-invasively. So we can have this kind of more ideas of diversity among bats, among population of bats, and what is important among seasonality, among season of bats. The immunological systems and the immune response of bats are better explored, thanks also to the people that are doing immunogenetics, but also immunoecology. I have no time really to develop all the immunoecological stuff, which were actually quite developed in bat ecology. So we know that bat immune system response are modulated according to many factors, and especially season and reproduction. I think we can now better relate this immune system, this immunoecology with the parasite diversity and with the bat ecology. So now I think we can really things to develop. It's time, really timing to develop field immunoecological studies. We can do, and Southeast Asia is a good place to do that now with this. So we can do that in Indonesia, we can do that in Philippines, we can do that in Indochina. So we can have several biological range. We have bats that are common species, but bats that are sometimes specific of each places with different habitat, with different changing progress, and we can do that really with all the new technologies that are available, virome, full genome sequencing, full proteome, and we can really do that, what I name immunoecological studies. Why it's interesting? Because it's difficult actually when I listen to our previous speakers, how to do and what is the cost, what are the costs to do surveillance, how to do biosurveillance of wildlife, what are the costs of doing this? It's quite very costly. We have to target the biosurveillance, we have to target the species, we have to target the place, the location, the place and the time. Developing field immunoecological studies and comparative analysis will allow us to know where are the likely place and the likely time for viral transmission, which is disease ecology, and after finally to have more ideas of spillover risks for other species and livestock or humans, and finally to complement and better complement the public health and the animal sectors by doing a better surveillance system and maybe less costly and more focused. Thank you very much, and thank you again for this invitation.
Okay, Serge, thank you so much. You raised a nice talk and nice discussion. Maybe some of us will have a new view about the immunogenetic in the wildlife. So you mentioned about why so many parasites. This is interesting because parasite can live so long in the animals as well as in the mammals, and they have a living strategy how to feed from the immune system of the host. This is interesting to see. And you mentioned also about ecological studies. This is very interesting because this is new for us. With metagenomic analysis and also with viriome and also with proteome analysis. This is new for all of us. This is time for discussion. I cannot summarize more than that because you raised a new point in this webinar. So Putu or Esel, how many questions already in the Vision Hall?
We have currently 6 questions.
Yeah, please, Esel, you can share the
I'll
question
share the
from
screen.
Pak Joko, yeah.
All right.
Esel is one of our students helping us to this.
Okay, for the first question, we have from Farida Hadayani, and it's towards Pak Joko. The question is as follows. Dear Pak Joko, thank you for your presentation. You have mentioned about the zoonotic diseases originated from bats, viruses, diseases. Are there any potential bacterial diseases that come from bats as well? The floor is yours, Pak Joko.
Okay, thank you. And I know Ibu Farida very well. So thank you for the questions, Ibu Farida. Well, potentially, yes, there are some bacterial disease that can be transmitted from bats to human. Yeah, but we don't know exactly like most of the bacterial disease probably could be transmitted from either animal, I mean, not just bats, but also other animals, yeah. And that is because more of the hygiene situations. So bacteria probably has more broader species of susceptibility, yeah, host range, yeah, compared to viruses. We understand that viruses will need some receptors, specific receptors, that will enable them to infect these species of hosts. With bacteria, basically, we could have even from not primate animals, non-primate animals, yeah, that can be transmitted to human beings. So still a lot of studies also conducted and probably done in more detail about why this bacterial has more broader range of host infections. But one of this is because they don't need quite specific receptors compared to what virus needs. I hope that answered the questions, Ibu Farida. If not, you can maybe unmute yourself and we continue or with a follow-up discussion.
Maybe if you have a response, you can type it in the chat section of the Zoom. Okay, we'll go to the next question if there is no response.
Okay.
It's towards Pat Joko again, and it's from Delwin Chapuyo from the Philippines. And it reads, "Good day, Dr. Joko. Thank you for your insightful presentation. May I know if there's an explanation why does coronaviruses need an intermediate host before it can be contracted by humans?"
Yeah, these questions I may not be able to answer precisely, exactly, and I probably will need help from Dr. Serge, yeah, in making this complete. But my understanding, yeah, compared to the other viruses like Nipah and rabies, they are in a state or in the stage where they are actually also infectious to humans, and they are in the same stage where they are harbored by the reservoir. So when they are in the reservoir, like bats, both Nipah and also rabies, they actually are ready to be able to infect human too. It's just a matter of how the interaction is going to happen, whether through something that may uh, occur directly, or they may also go through other animals. So although those two viruses are known to be able to be transmitted directly, but still in many cases, the infections in human also occur through the intermediate host. But the fact that in Bangladesh, some studies by the CDC scientists that put the infrared camera during the nights, the bats actually are urinating to the sap, to the dead palms that are harvested by the local people. And then this raw dead sap actually are consumed by the people, and uh, they could infect the people right away without any involvement of the intermediate host. Rabies, I, I cannot tell exactly the cases of rabies um, um, transmission directly to the humans, but there was one report, I have to actually read that again, in the US, basically, that could be potentially that humans are getting the rabies from bats infected directly. but the paper was probably from a long while ago, so I have to dig that data again. That is the thing so far I may be able to explain, but if Dr. Serge could help me with the answer, please.
Thank you very much. Yes, it's a complicated question because we don't know exactly what happened, but what we can say for the moment for all of this group of beta coronaviruses in bat and actually they don't have the receptors that is the key to enter in the immune cells of humans immune cells. So they need to have the key to go to the lock of this. And which is, we don't understand exactly how they got this, but they can get this by passing to other mammals, and especially the carnivores, which are quite good, but not all carnivores that quite good, but some other one, where they could mutate, or they could occur, or they could also exchange these and get this key from other coronaviruses. So it's a way to make this. And after, when they have got the key receptor, and especially the spike receptors that are doing the spike of the virus, they could really like a key to enter in the receptors and enter in the human cells. What we don't understand is why. But when you look, the bats are quite very far from the humans. So it's maybe also explaining that they need also to jump finally to something that is close to humans. And sometimes it could be the livestock. Most of the time could be the pigs, could be the camels, could be the horses, and could be the carnivores.
Thank you,
Thank
Dr.
you for the
Serge.
response, Dr. Joko and Mr. Serge. We'll go to the next question. It's towards Dr. Jose. Read from Farida Hanadayani. It says, Dr. Jose, to laboratory tests for rabies in bats, did you use a specific primer, PCR, for classic rabies or Australian bat Lisa virus, ABLV? Because there are in the same group of viruses. Thank you. Maybe Dr. Jose, you may answer.
Okay, thank you for that question. What I cited a while ago is the activity done by the Department of Health in the diagnosis of rabies, especially and the Department of Agriculture doing the brain examination of the dogs. What I know is what is widely used in the Philippines is the demonstration of the inclusion bodies, and not much on the PCR thing. I'm not very sure of what they're doing at the national headquarters for diagnosis. Thank you.
Okay, thank you for the response, Dr. Jose. And we'll go to the next one. It's back to Dr. Joko, and it's from Ershandi. Dear Dr. Joko, how much the danger of bats could arise the IED if confirmed with the other animals in the future, and how much damage the IED for the country, especially in Indonesia? Thanks.
Hey, thank you for the question. Is this Miss or Mr. Ershandi?
From Lombok.
Yeah. So basically, yeah, as I mentioned, I think I put some information on the slide about the importance of having triangulated surveillance conducted in wildlife, livestock, and also human beings. So this has to be done in the approach of One Health, of course, yeah. And we have tried these efforts in 2016 through 2019, but the problem with this concurrent surveillance, we have to have like a lag of time if one is doing the wildlife surveillance So then the livestock surveillance must have some intercept time not longer than 2 weeks before the start of the wildlife surveillance, and not later than 2 weeks after the wildlife surveillance. So basically, we have to put some efforts in doing the 3 surveillance. Then the timing schedule must be organized, and also the area we have to have some agreement. Yeah. So within the radius, or the the radius of surveillance, certain kilometers. For instance, if we put the surveillance of wildlife into one point, so we could only do the livestock surveillance and also the human surveillance in a certain diameters or certain radius. So basically, we had to agree on several factors like the time and also the area. And from our studies, we have not been able to conclude. We still have to put some analysis onto that data, but we haven't done so. But other things that we also see about the importance of the surveillance in wildlife, livestock, and human beings using the PREDICT protocol. I mentioned about the non-specific or more broad spectrum or degenerate PCR, the consensus PCR. So in the past, the livestock labs, they try to identify certain diseases that has been showing in the environment, and they were not able to identify the agent, the pathogens that cause the diseases. And using this consensus PCR, they were able actually to see the correlations of the disease and the causal agents. And this, of course, will have some impact, very important, that using this degenerate primer PCR is able to detect not in a species level, but also in the genus level. So very important to also include a larger area. What we have done in Sulawesi is only certain areas in 2 provinces, West Sulawesi and also North Sulawesi. So basically, it will be required to cover larger areas of the country, also including the outside of Sulawesi. But
Okay.
that is the answer that I can give you.
Thank you for the very clear response, Dr. Joko. Okay, we'll have to go to the next one. And it's towards Dr. Joko again. It's from RR Upik, and it reads, Dear Dr. Joko, thank you for present research and presentation. How about the new findings? Is there all types of species that becomes the reservoir? Because there are different types of bats, for example, fruit bats and insectivore bats.
Yeah, thank you again for the questions, Roro Opik. Yeah, we realized that we have some limitation in our studies, that we only survey bats with big size, macro bats, which is the Theropus and also Acerodon genus. So basically, we did not sample any other kind of bats in a smaller size, whether they are insectivores or they are maybe some fruit eater bats. Yeah? But with this limitation, we cannot actually say that we have some safe situation in Indonesia, because we only limit to 2 species majority. While we understand that there are lots of other genera and the other species bats in Indonesia. And as I mentioned also in one of the slides that the Rhinolophus species actually are also can be found in Indonesia, in Sumatra, Java, and Kalimantan. So we didn't survey that, yeah. So because of the driver that we studied is about human behavior, especially the consumptions of wildlife. So we only limit to these two species that are actually the favorite species of bats in North Sulawesi to be consumed because of the size. So once again, although we did not find any alarming results or findings in the lab, but it doesn't mean that we are in a very safe situation because we have to actually cover more species of bats to be surveyed. Yeah,
Okay,
over.
thank you. Okay, we'll have to rush because we're gonna end at 15:15. So this one is towards Dr. Morand from Siargao Princess. Hello, Dr. Morand. In
Hello.
comparison between bats and rodents are reservoirs for zoonotic disease viruses, you stated that the birth frequency
Yeah.
in bats have less immunocapacity causing viremia. What do you mean by that, doctor?
Thank you. This is, yes, it's right. I did not have enough time to explain this. Okay, what we know that our immune system or immunocompetency is modulated and is modulated also according to our physiology. And especially we know that during a mammal pregnancy for all, including humans, when the females and the women are starting to go to pregnancy, usually there is a decrease of the immunocompetency, the immune system. Why? Is mostly also to protect the baby and inside the starting of the baby and after really the baby. So there is this decrease. Even if there is also the help of the mother that can share some immunity with the baby inside the womb. So for the bat, it's quite important because we know that, for example, for the Nipah virus, so the Nipah virus, that is also modulated during this, most of the spillover to humans or to animals occurs during the reproduction season. And so during the reproduction season, actually the immune system is really dropped. So there is no control of the virus. So there is starting of the viremia, again, starting also to interaction, starting to the spread of the virus. So there is increase of the prevalence on viremia in bats. So it is mostly at this time of the reproduction of the bats that there are the most risk of transmission, spillovers, and the risk of infection if humans. We have also some works done by colleagues, and they found the same thing in bats in Europe. When we look at the ectoparasite of bats, is also you can modulate this with the immune modulation. The ectoparasite abundance change with the investment in the human systems. And when there is a reproduction, pregnancy, against decrease of the immune system, no control of the parasite, increase of the parasite.
Thank you for the response. We'll go to the next question. And it's towards Mr. Moran again. It's from Nurul Hidayah, and it reads, Dear Mr. Moran, how's mechanisms immune response in bats that could infect to humans? Is it the same receptors in bats and humans? Thank you. I believe you kind of answered this in the previous question.
Yeah.
Because it was because of the intermediate host.
Yeah, exactly. So this is exactly related to this, but also it's related to what we need to do more is a comparative immunogenetics. We really need to better compare. For example, why finally the livestock are so important. Dr. Joko, but also Dr. Jose show us before that some animals like the poultry, like the pigs, like the horses, like the livestock are very important in sharing viruses. Why? Because if the viruses or the bacteria is going there, they can humanize, they can better know, they can better adapt. And finally, because we share some commonness in terms of immunogenetics between us, because we are sharing the same communities of pathogens, we are same communities. So we need to do more about comparative immunogenetics.
Okay, thank you for the response. And I believe we have 4 more minutes, so we'll have to rush through these last 4 questions. So this is towards Dr. Jose, and it reads, is it necessary or not to regulate the prohibition of trading bat meat since there are zoonotic diseases that originated from bats? So more on the trade of bushmeat, if Mr. Jose can respond. On Mr. Morand, sorry.
I'm not very familiar with bat trading because we don't trade bats in the Philippines. There are poachers who do this.
Yeah, I think it's a good question, but it was already a little reply that saying that is maybe who are the person at risk. And I think the problem of bat meat, or the problem of I would say illegal selling, illegal importation, all of this related to bats and other wildlife is environmental crime. So it's something this. For me, what are the number of people that are really infected by this? So we have to really to know, working with the meat, and especially the meat of wildlife, and what is the importance of the really increasing of the wildlife that are put in farming now. There is increase of farming of wild animals, and civets, pangolin. Now we have also the raccoon, also this the vivarium dogs. We have a lot of this, especially in some countries, which with increasing with the farm, with mostly other animals, with condition that could be put the animals at stress, we maybe create these things. So I would like to really now to focus on this problem of the white farming.
If I may add something a little. Yeah, I agree that we have to be in a spirit in stopping this wildlife trade, yeah? Not only bats and rodents, but also other wildlife. And this has been going on also in efforts by the authority from the Indonesian side, like the Ministry of Environment and Forestry, as well as other related ministry like the Domestic Affairs Ministry as well. But it has to be, what do you call it? We have to actually make an echo, yeah, of this activity, not only from the top at the center of the policy maker, but also to the grassroots level at the local level. And this has to be done in coordination, so that a lot of activities actually could be, potentially could be stopped. But since there are no actual sanction, there are no actual punishment in the grassroots level, so people are still doing that. So that is one practical answer to this question from Ibu Ratna Hartanto. Thank you.
May I add for that? May I continue? In the Philippines, we have already sets of regulations. This is illegal, and there is an equivalent punishment for trading wildlife species in the Philippines. But there are a lot of poachers, and this is our problem. And we don't have enough authorities to implement such law. Thank you.
I believe our time is up, but there is
Actually, we
three
can
questions.
maybe for next year something discuss about maybe on how to implement a better way to work the conservation, the police and the regulation. I know this is also a problem I discussed with the French and the French control for the border control, and they're also afraid about and they have a lot of illegal importation of wildlife, and they want to, and maybe it's time also maybe to put the people for the public health concern, the conservation, the animal health concerns, and the wildlife trafficking, but also the natural way with the community, so the natural way to still use the wildlife. So maybe next year, Professor Wayan, we can organize something.
Okay, thank you, Esel, for sharing the question to all the participants. But I still have a curiosity to know, Serge, that so far I know that bat have a lifespan until 40 years. So you mentioned with the diversity of viruses, diversity of parasite, and diversity of ectoparasite in bat. Is that long their life, they can have as a reservoir for the disease?
Yes. Alors, there are some other animals that live very longer also that are not very so much diverse, but it's one of the hypotheses, yes. Yeah. Longevity, but also to save energy for the flying, for this longevity, for the stress that may be, for example, they have also a very high metabolic rate, and all of this costs a lot of energy, and maybe the bats also, they save this energy at the expense of the immune system. That's why. So maybe the immune system is different. They have to tolerate much more the parasite than maybe other animals. So maybe we have to look also at the tolerance of bats to the virus and the parasite diversity compared to other animals.
This could be because of the low temperature of the bat in compared to the human being on other mammals, because the normal temperature is below 35.
Yeah, it's a low, but with a high metabolic rate, which is quite impressive. The metabolic rate is really higher than all other mammals. So they are very, very special. So complicated animals finally.
Okay, thank you guys. Thank you, Dr. Joko. Thank you, Serge, and thank you, Jose. This is a very interesting discussion for today. But I inform you that the participant is around registered 520, but they joined it through Zoom and through Google, through YouTube. Yeah. And we attendants, they come from Indonesia mostly, and also from Uganda, from the Philippines, from Thailand, from Malaysia, and from Hungary, and others, some other countries. This is interesting to discuss with all of us. We still have another, in 2 days from now, we will continue with the tool that how we can identify the pathogen in bat, in wildlife, through using the modern technology, the latest technology, microbiome analysis, and also metagenomic analysis. Maybe Putu can introduce what we want to know in 2 days from now. Thank you again, Pak Joko and Serge, and also Jose.
Thank you.
Thank you so much for participation.
Thank you for the organization all the time. Very good organization, very good group. Thank you very much. Thank you for the question. Thank you for the participant.
Nice meeting you, Dr. Serge and Dr. Jose.
Thank you very much.
Online,
Very good.
Putu, please.
Thank you, Jose. Bye-bye. Bye-bye.
Please don't say bye-bye first. We need to take a picture together. Before I close the session, I would like to say thank you very much. Oh, okay, Dr. Serge, maybe already go. Okay, that's okay. Thank you very much for Professor Wayan. Thank you, Ezel, for helping us to conduct the question and answer and also the session. And also thank you very much for all of the speakers for your awesome presentation. Today we learned about the occurrence of the wildlife origin zoonotic diseases and the possible transmission, the evolution that can be held inside of the wildlife, and also we learn about the immunogenetics. And maybe before we finish this webinar, I would like to invite all of the panelists. Yeah, maybe Pak Heri and Mbak Della can turn on the video before I share the the attendance form. Okay. Sorry for our selfie.
If I may greet Pak Heri, selamat sore, apa kabar?
1, 2, and 3. Once again, 1, 2, and 3. Okay, thank you very much. I will share for the participant. We will share the attendance form, and maybe Noli and Sasrin can help me to share the link to the chat box. So
thank you for our panelists.
Yeah.
Wonderful presentation.
Thank you, Pak Heri.
Yes, sama-sama Pak Imung. Terima kasih Pak Imung.
Oh, dulu Pak Heri depan Pak Imung ya.
Baik, teman baik ini, gelundang-gelundung ini, Prof.
Pantesan. So Pak Heri adalah staf kami di OHCC, Prof.
Oh, good
Pak
to
Imung,
hear that.
Ya. Kita ada makaka juga ekor panjang ini. Nanti minta bantuan juga ke Dokter Imung nanti. Oke, thank you so much. Kita masih ada 2 hari lagi kita untuk ke metodologinya. Terima kasih Pak Joko.
Terima kasih.
Terima kasih saya juga untuk semuanya.
Terima kasih
adik-adik adik-adik panitia juga.
Iya, ini jangan bosan-bosan loh Pak Joko bantu kita.
Saya izin pamit ya.
Iya, sama-sama Pak Joko.
Terima kasih Pak.
Selamat sore.
Selamat sore.
And for all the participants, don't forget to join our second webinar. We will talk about the novel technology for wildlife pathogens identification. As we ever heard from Dr. Serge that he ever mentioned about the NGS, the next generation sequencing, the bioinformatics, and everything about the nowadays technology we can learn and discuss together on the upcoming 2 days. So please make sure, mark your schedule, mark your calendar, and we will meet on the 2 days ahead. We will wait for 10 minutes until you finish to fill the attendance form, and then I will stop share and close the Zoom application. Thank you very much.